Okay, so we're talking about glycosidic bonds. So remember that our disaccharides are formed through a condensation reaction. So we have an individual monosaccharide plus another monosaccharide, and we get a disaccharide, right? And then we also produce water in each of these. So how does that actually happen? So remember, all sugars are coated in hydroxyls. And so if you look at the anomeric carbon, so finding the anomeric carbon is the carbon that's bonded to 2 oxygen molecules, right? So we found our anomeric carbon, and you're going to look at the hydroxyl associated with that anomeric carbon. We're going to take away that hydroxyl, and then on the next sugar, we're going to take away a hydrogen from one of the hydroxyls, and we'll tell you which one it'll be. So in this case, here's my anomeric carbon. So this is carbon number 1, 2, 3, 4. So it's the hydroxyl on the 4th carbon. So what's going to happen is that I'm going to take away the OH and the H. So remember that makes HOH, and HOH is the same thing as water. So if I take away these things, now this carbon, this anomeric carbon right here, is going to make a bond with that oxygen that's on the second monosaccharide. And so what that ends up looking like is this. And so we form this new bond. This is the new bond that we drew right here, and that's the same new bond that's right there. So this is the glycosidic bond. So the question is, right, when we form any glycosidic bond, we have to name it, because the way that the glycosidic bond forms tells you about its strength, tells you about how that disaccharide or polysaccharide is going to behave. And so if we look at what we started out with originally, kind of erase all this stuff. Right, okay, so now if we look at what we started out with originally, each monosaccharide starts out as a particular anomer. So either it is the alpha anomer or the beta anomer. So if I look at this one, I find my anomeric carbon, and there's no other carbon attached to it. So this is carbon 1, 2, 3, 4, 5, and this is carbon number 6. So if I look at the hydroxyl that is attached to the anomeric carbon and the largest numbered carbon that is outside of the ring, and I compare their orientations, this one, because it's going, carbon 6, because it's going up, it is above the ring. So we're going to call it up. The hydrogen over here, right here, is down, right? So that's how you can tell up and down. So the carbon 6 is up. The hydroxyl here is going down. And so if you have something that is up and down, we call this an alpha anomer. So if you have an alpha anomer that creates a glycosidic bond right here, so this is my anomeric carbon, and I had an alpha anomer to start with, I'm going to have an alpha glycosidic bond, and I can check that. I can say, okay, where is the oxygen in the glycosidic bond? Because it's no longer a hydroxide. It's now just an oxygen. And in this case, we're going down, right? And here we are going up. So up and down is going to be alpha. So the next thing that we have to do when we name a glycosidic bond is not just say, is it alpha or is it beta, but also to say, what is the position of which carbons are participating, which carbons are participating in the glycosidic bond. So in this case, we have an anomeric carbon on the first monomer, and there's no carbon attached to it. So that's carbon 1, 2, 3, 4, 5, 6. I like to number them all. On my second carbon, here's my anomeric carbon because it's attached to 2 oxygen molecules. So I'm going to give it carbon 1 because there's no other carbon attached, 2, 3, 4, 5, 6. So the 2 carbon atoms that are participating in the glycosidic bond, if I can kind of erase some of this stuff that's going on. So the 2 that are participating are this one and this one. So that first one is carbon number 1. So we're going to call it a 1, and the second one is carbon number 4 that's participating, so we call it a 1,4. And then any anomeric carbon, so this is an anomeric carbon that participates in a glycosidic bond. This anomeric carbon is not participating in a glycosidic bond, so we do not include it, right? But this one is participating in a glycosidic bond, so we include it, and we're going to call this an alpha 1,4 glycosidic bond, right? So if you kind of go along, these questions kind of help you to answer those. So in figure 1 are the glucose molecules that are reacting in the condensation shown as the alpha or the beta anomer. Well, the first one is an alpha anomer, right? But we didn't talk about the second one. So let's look at the second one. The second one, we have a C6, and we have our hydroxyl attached to our anomeric carbon. And so this one is up and this one is down. And that means this is also the alpha anomer. And that's just a way to write alpha. So they are both are alpha. Okay. So which carbons are bonded in the glycosidic bond in D-maltose? So here, this is saying a disaccharide containing 2 glucose units, which is called maltose. So in D-maltose, the carbon number linked on the left glucose molecule is, let's see if I can highlight a different color. This is my left glucose molecule, number on the left. And the one that is participating in the glycosidic bond, we said, was number 1, right? And then the one on the right, let's do a different color. This is my one on the right. So this one on the right, the carbon that's participating, we said that was carbon number 4. So 1 and 4. So do you see why a glycosidic bond in maltose is called an alpha 1,4? And that's because, right, on the first saccharide unit, we no longer call it a monomer anymore. So on the first saccharide unit, C1 participates, participates in the glycosidic bond, while, right, C4 of the second saccharide unit participates. Okay. And then the other thing we have to say is, why are we only looking at the orientation of one of the monomers? Well, we're only saying 1 alpha, right? We don't say alpha alpha, because if I look at this, okay, here's my hydroxyl and here's my carbon, so this should also be alpha, but I don't say alpha alpha 1,4. That's incorrect. I only say 1 because it only has 1 anomeric carbon that participates in the glycosidic bond, right? This anomeric carbon does not participate. And only the anomeric carbon on the first unit participates in the glycosidic bond. Okay. All right, so now you try to name the glycosidic bond in all of these. Take a minute, pause the video, and then I'm going to do it. Okay, so here, the first thing that I always do is find my anomeric carbon. Here's my anomeric carbon. Here's my anomeric carbon. How did I know that? I know that because the anomeric carbon is bound to 1, 2 oxygens. This anomeric carbon is bound to 1, 2 oxygens. So that's how I know it's an anomeric carbon. Now I need to go in and I need to number my carbons. So if I number my carbons, the anomeric carbon gets carbon number 1 unless there's a carbon attached to it that is outside of the ring, and that is not present. So I go 1, 2, 3, 4, 5, 6. We'll do the same thing over here. 1, 2, 3, 4, 5, 6. So now I need to name the carbons that participate in the bond. So the carbons that are participating in the glycosidic bond are here's carbon 1 and this carbon 4. So I'm going to call this a 1,4. Now, any anomeric carbon that participates in the glycosidic bond, I need to say if it is an alpha anomer or a beta anomer. So I only have one glycosidic bond. Let me erase these. I only have one anomeric carbon that participates in the glycosidic bond. The glycosidic bond is right here. And this is the only carbon that participates. This one is not participating in the glycosidic bond, so we do not include it, right? So I need to look, and I'm going to say, okay, my C6OH is going up, and then my glycosidic bond is going down. So up and down is alpha. So that's an alpha 1,4. All right. Now, do the same thing. Find your anomeric carbons. Found my anomeric carbon because I'm bound to 2 oxygen atoms. Find your anomeric carbon, bound to 2 oxygen atoms. Number your carbons. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Okay, so the 2 carbons that participate are carbon 1 and carbon 4. So again, I call this a 1,4. Now, you have to look at the anomeric carbons that are participating. This is an anomeric carbon in a glycosidic bond. This anomeric carbon does not participate in a glycosidic bond. So I only need to worry about this one. So I look here and I have CH2OH, C6 is in the up position, whereas my glycosidic bond now is also in the up position. So up and up we call beta. So this is a beta 1,4. All right, now this one is numbered for you, which I will not do on the exam. But if we look at this, let's do the first one. Let's do the top one first. I find my anomeric carbon, which is bound to 2 oxygen atoms. I'm going to number it 1 as long as there's no other carbon attached to it outside of the ring. 2, 3, 4, 5, 6. All right. I'm going to do my second one. I'm going to find my anomeric carbon. In order to find my anomeric carbon, it's right here because it's bound to this oxygen and this oxygen. But be very careful because this anomeric carbon on the sugar at the bottom has a carbon attached to it that is outside of the ring. So you have to give this carbon number 1, which makes the anomeric carbon number 2. Then 3, 4, 5, and 6. I will not number these on the exam. So please, please, please get your numbering. So now what I notice is, oh my goodness, the carbons that participate are carbon 1 and carbon 2, but both of those carbons, both anomeric carbons participate in glycosidic bond. Glycosidic bond. So that tells me I'm going to have 2 letters like alpha alpha or alpha beta or beta alpha, whatever it is, right? So, okay, let's do this just like we did the other ones. What numbered carbons are participating? In the top one, it's carbon 1. In the bottom one, it's carbon 2. So put that in parentheses. Now, in the outside of the parentheses, do the top one first. I have my C6 is going up. My glycosidic bond is going down. So up and down tells me that that is alpha. Then on my bottom one, I have C6OH going up. I have my glycosidic bond going up and up, tells me beta. So this is an alpha beta 1,2. Okay. Let's see. Oh, the bonds just, the determinant glycosidic bonds, we did that. The glycosidic bond in D-sucrose is different, right? So this is D-sucrose. It's different because both anomeric carbons condense. So we did that. Label the monosaccharides in the sucrose as either alpha D-glucose or beta D-glucose. Well, so if we look, right, at this top one, if we look at the top one individually, right, if this one's up, the hydroxyl would have been here. It would have been an OH. So we could say up and down is alpha, right? So it would have been alpha. So the first one, label it. Oh, okay. So I have to label it alpha. It would be called alpha D glucose. Whereas the bottom one, we have an up, and then the OH would also be going up. So up and up gives me beta D glucose, right? Okay, so let's see, what else do we have to do? Okay, so that's number 5. Then why do the glycosidic bonds in maltose and lactose only contain either alpha or beta, and sucrose is an alpha and beta? So in maltose and lactose, right, they have only one, and that is because only one anomeric carbon participates in the glycosidic bond. Okay. All right, locate and name the glycosidic bonds in the disaccharides. All right, so pause it and you try it. First things first, I'm going to find my anomeric carbon. My anomeric carbon is bound to 2 oxygen atoms. I number my carbons, 1, 2, 3, 4, 5, 6. Okay, I'm looking for my greatest number that's outside the ring, and I'm going to compare that to my glycosidic bond. So this one's going up, this one's going up. So up and up tells me that that is beta. Now I just have to say which carbons participate. So my other sugar molecule, I'm going to look, here's my anomeric, bound to 2 oxygens. So that's 1, 2, 3, 4, 5, 6. So do I need to look at up and down on this one? No, because my anomeric carbon is not participating. So all I have to do is go find which carbons are participating in the glycosidic bond. So it's going to be the 1 and 4. So this is called a beta 1,4. The next one. Okay. So here on my first one, here's my anomeric carbon bound to 2 oxygen atoms. I'm going to get this 1, 2, 3, 4, 5, 6. I look here, this is my 6, here's my anomeric. So this one's going up. My glycosidic bond is going down. Up, down is alpha. All right, so now I go to my next sugar. Here's my anomeric carbon bound to 1, 2, gets the number 1, 2, 3, 4, 5, 6. So which atoms are participating? Carbon 6 and carbon 1. And we usually read left to right and up to down. So it'll be a 1, 6, an alpha 1, 6. Okay. So number 8, in your own words, write out how to determine glycosidic bonds and disaccharides. Okay, so that I'm going to leave on you. Identify each of the reactions as either condensation or hydrolysis and provide the products. So in this reaction, I'm taking a disaccharide and I'm going to use water to break it up. So this is going to be a hydrolysis, right? And so I'm going to cheat over here. And I'm going to take the disaccharide. I'm going to show you an easy way. I'm going to paste it kind of down here because I don't really have enough room, right? But here are my products. So what do I have to change? Well, see if I can just kind of erase what I need to erase. Okay. So the only thing that's going to be different is over here on the glycosidic bond. So I'm going to erase what links the two together and then redraw what should go on there. So when I have water, right, I have HOH, right? And then there's an oxygen here. So one of these is going to keep the oxygen that was there. So we are going to give it to this one. And we're going to say, okay, you can keep the oxygen. Now, the rest are going to pick up the parts from water. So I'm going to break the water, not in half, but I'm going to break the water so that I have a hydrogen that's going to be attached here, and then a hydroxyl that's going to be attached there. And so this is maltose. And so since we start out with maltose, I know maltose is 2 units of glucose. So this is D-glucose, and this is D-glucose. And you could actually even go even further and say that since this is up and this is down. This is alpha D glucose. This is up. This is down. This is alpha D glucose. Okay. All right, let's do the next one. We have galactose and D-glucose. We are adding them together. So if we add these two together, we're going to have a condensation reaction because we're going to make water. So I'm going to show you how we do that. So I'm going to take these reactants. I'm going to paste them. Okay. So here are going to be my products. And I'll kind of do my little erasing trick. Okay. So get rid of that. So what's going to be different is what's at the glycosidic bond, right? So actually, if I could do this, I actually want to do these individually. Crop. So we can do this. Okay. And then I'm going to paste again, and I'm going to get the other half. Okay. So there we go. So now what's going to change is where the oxygen is going to be, right, on that anomeric carbon. So here's an anomeric carbon, and here's an anomeric carbon. Well, what's going to happen is that on that anomeric carbon, we have a hydroxyl, and then we're going to bind that to another hydroxyl that's on the other sugar molecule. So let's do it on this one. So if we do that, what's going to end up happening is that we're going to lose, right, a hydroxyl from one and just the hydrogen from the other. So if I remove those two, what's left? So I'm going to use my trick, right, and remove them. What's left? Now what I can do is I can just kind of link these together. Oh, it doesn't want to let me do that. Let's do this. And then I'll go in. Oh, it doesn't want to let me do it. There we go. Okay. So now I'm getting rid of that hydrogen. And that's just the number one, but you don't really need that. So now I'm just going to, link them together in the glycosidic bond. So all I did was I took away an H and then an OH, and I made water. So I made, this is actually D-lactose. D-lactose is a galactose and a glucose, right? And that's it. That's easy. Okay. Circle the glycosidic bond in the following carbohydrates. So it's always going to be between one of the anomeric carbons. And so if we pick the two anomeric carbons, there's only one anomeric carbon that participates in a bond linking the two sugars. And that is right there. Here's one anomeric carbon, and the other one doesn't have an anomeric carbon. So it has to be that one right there. All right, so we have a trisaccharide. Ooh, cool, a trisaccharide. So we need to name the glycosidic bond between monosaccharides A and B. Okay, so I find my anomeric carbon. Let me do a different color. So I find my anomeric carbon because I'm bound to 2 oxygens. So this is carbon number 1, and it's numbered for you. I would not do that on test, but that's carbon number 1. Here, the carbon that's participating in that double bond is actually right, it's not the anomeric, but is right there. It's this carbon that's participating in the double bond. So here's my anomeric carbon because it's bonded to 2 oxygen atoms. So now if I want to number this carbon right here has to be carbon number 1. So my anomeric carbon is carbon number 2. So then the one participating in the bond is carbon number 3. So between the two, A and B, I have a 1-3 bond. And now I need to say, is this alpha or beta? So I look here and I go, okay, this one is up. And then here, the one that's bonded to the anomeric carbon is going down. Up and down is telling me it's alpha. Name a glycosidic bond between monosaccharides B and C. All right. So now I kind of have to switch gears, right? And I have to say, okay, here's my anomeric carbon because I'm bound to 2 oxygens. I know that this is carbon number 2. Then I'm going to go to my other sugar molecule. Now be careful with this. Here's my anomeric carbon because it's bonded to 2 oxygen molecules. You see it's kind of flipped. So that's a 2 to 1, right? It's a 2 to 1. Now, is this alpha or beta? Well, look at this one. See the C6 that's outside, this is going down. It's different than the normal written way, but all you have to do is compare this to the glycosidic bond. And so we actually have two molecules that are 2 anomeric carbons that are participating in the glycosidic bond. So in molecule C, we have the CH2OH going down, we have the glycosidic bond going up. So for that second sugar molecule, if you go up and down is alpha. If we look at the B molecule that's participating in this glycosidic bond, we can see that here we have a C6OH, this is going up, and we have a glycosidic bond that's going up. So up and up is beta. So we have a beta alpha 2,1 glycosidic bond, right? Then it says the trisaccharide contains fructose and glucose, label glucose and fructose. So A and C, you can see that these are 6-membered rings, right? Because a 6-membered ring looks like this. This is a 6-membered ring, and then we know a 5-membered ring looks like this. So we know that glucose is a 5-membered ring, and then we know that fructose is a 5-membered ring. So we can say that monomers A and C are glucose, and then we know that monomer B is fructose. Okay,